For the rest of the session, we have 4 short talks and then a panel discussion. And it's my distinct pleasure to introduce my co-moderator to give the first talk and maybe a word or two about the introduction to the session. George Patrinos from the University of Patras in Greece will tell us about the Go Greece project. George.
Yeah, so before I start, I would like to thank the G2MC team, particularly Teji, Hannah, Meredith and their team for putting together such a really professional meeting, a virtual meeting. It was really a last-minute decision to move to hold this virtual meeting after the COVID-19 pandemic. So it has been a major undertaking, and they really did a fantastic job in assembling all this material together. So, as Alan said, in the next 10 minutes, I'll try to squeeze a lecture about a national initiative we have started 10 years ago in Greece, and we call it the Genome of Greece, or the Go Greece initiative. So next slide, please. Next slide, please, so that we can kick off. That's one of my favorite slides, my favorite locations in Athens. That's, of course, the Parthenon. And I frequently use this as a paradigm of how we could use this temple to mimic the transition from genomics research to genomic medicine. And if you go to the next slide, please, you can clearly see that we can start by having the genomics research at the ground, at the foundation of the temple, and reaching to the personalized medicine, which lies at the roof of the temple. So to move from the foundations to the roof, we need pillars. And each one of these pillars correspond to different disciplines that they actually drive this transition from research to the clinic, from the bench to the bedside. And that includes capacity building, ethics, informatics, economics, raising health care professionals' education, as well as increasing the genetics awareness of the public. So all of these are interesting aspects that one needs to address when he or she touches upon the concept of large-scale genomics initiative. So next slide, please. There are several national-wide large-scale genome projects. We heard earlier today a nice presentation about the DNA do Brasil, which is a very nice undertaking in Brazil, one of the major Latin American countries. Next slide, please. So the Genome of Greece is a large-scale initiative that's focused on how you could implement personalized medicine in Greece. Next slide, please. Okay, so the goals is to determine the allelic architecture of the Greek population, focusing on rare, pathogenic or not variants that are characteristic for the Greek population, to characterize the unique allelic spectrum for isolate populations in Greece, which are many, and we're talking the islands, the mountainous region, as well as subpopulation groups such as the Greek Roma and the Pomaks. Another one is to establish the pathogenic variant spectrum of common as well as rare genetic disease in the Greek population, to catalyze the clinical implementation of pharmacogenomics in the Greek healthcare setting. Next slide, please. How to develop and maintain a comprehensive knowledge base to report the genetic heterogeneity of the Greek population as far as pathogenic variants and polymorphic alleles are concerned, to address the ELSI issues, the ethical, legal, and societal issues that pertain to personalized medicine, to enrich the genomic awareness of the healthcare professionals as well as of the general public, which is, of course, fundamental because this also touches upon ethics. Next slide, please. Okay, so this project constitutes of 10 work packages. One is the government side coordination, and with dark blue, you also see that this also touches upon dissemination and communication. That also goes hand in hand with fundraising. In light blue, you can see 4 different work packages that has to do with sample collection, genetic analysis, genome informatics, as well as biobanking. With light red, you see that one important component is the ethical, legal, and societal issues, which also includes the stakeholder analysis. We need to ensure we should understand how the stakeholders think about this particular discipline. Then we also have the pricing and reimbursement and the regulatory aspects for implementing personalized medicine, which means economic analysis, economic evaluation, pricing, and reimbursement. And then lastly, we have the dark red, which includes educating healthcare professionals, as well as engaging general public in all aspects of personalized medicine. Next slide, please. Let's see now some numbers. The project kicked off in 2010. That means 10 years ago. And that actually, I felt that presenting some preliminary findings in the nice in the nice weather of Santiago would be a nice opportunity. Nevertheless, we can do that next year. The first Greek that has been sequenced, and that was thanks to Complete Genomics and Rade Darmanak, was in 2012, in July 2012. That was actually the first Greek sequenced overall. And we expected, according to the initial design of the project, that by this year we're going to have the first 1,000 Greeks sequenced, not necessarily by whole genome, but also by whole exome or by using clinically relevant panels. Next slide, please. The project goes so far in 4 different centers. The coordinating center is in Patras, in our laboratory, in Athens, in Thessaloniki, and in Crete, which is basically a center that coordinates the isolation of DNA samplers from a particularly interesting isolate population, which is in a mountainous region in Crete, Anoia. Next slide, please. So far, we have recruited more than 1,500 samples. We have sequenced by whole genome sequencing 61, by whole exome sequencing 109, and roughly over 700 individuals have been sequenced by using a proprietary panel for pharmacogenomic variants. Next slide, please. We have 9 projects initiated so far, 11 scientific articles produced, 5 master and PhD theses defended, and 4 national-wide conferences organized addressing both the public as well as healthcare professionals. Next slide, please. So let's go and see some of the preliminary findings. First of all was a paper describing a novel genetic locus that is associated with sporadic amyotrophic lateral sclerosis in Greek patients. Next slide, please. In which case we have adopted the stepwise approach, including a 3-tier sequencing cohort. The first one included whole genome sequencing of ALS patients, and then a validation study using two different independent cohorts. Next slide, please. Where we found that the gene that was involved in obesity, which was the FTO gene, expresses quite specifically in motor neurons, which is one extra functional evidence that this particular gene may be really involved in the pathogenesis of ALS. Next slide, please. Similar finding was the identification of novel risk variants for pediatric celiac disease in Greek patients. Next slide, please. Again, adopting a whole genome sequencing approach. That's a quite interesting undertaking, which is basically the determination of variants that are Greek-specific as compared to Caucasian population. Of course, Greeks are Caucasian, so we have used data from healthy donors, used the proprietary tool that we have developed in Patras. We called it Freaky for the calculation of the frequencies of variants. It's quite awkward name, as you can imagine. And as you can see in the next slide, which is a nice Manhattan plot, you can see that we have determined over 80 of those genomic variants that are very specific. They're, if I may say, they're Greek-specific, which may be used for different, applications downstream. Next slide, please. Then we went on to determine and critically appraise the views of healthcare professionals as far as genomics and personalized medicine in Greece is concerned. Next slide, please. For example, we found some very interesting findings. As you can see in question 9b, we asked the healthcare professional whether they believe that humans have 48 chromosomes, and 32% said yes. We also asked them whether adenine pairs with cytosine, and 13.2% said yes. So this clearly demonstrates that there is an urgent need to raise genomic awareness and education in healthcare professionals in the Greek healthcare community, if we're talking to implement genomic medicine in Greece. Next slide, please. You can see, of course, that they're very fair to understand that they do not feel qualified to interpret a pharmacogenomic test because they feel that the undergraduate studies did not provide them with sufficient knowledge on pharmacogenomics and personalized medicine. Next slide, please. And then we move on to determine how the general public thinks about personalized medicine. Next slide, please. And as you can see, the vast majority of the general public feels against direct-to-consumer genetic testing, and they really feel that physicians and not pharmacists should be the one to prescribe a genetic test as the healthcare professional that intermediates between the genetic laboratory and themselves. Next slide, please. This is a quite interesting finding because we asked the general public whether they believe that the personalized diet could be beneficial to them. And over 75% said yes. And that's quite a misleading issue that we can identify here because there is no such thing as genetically guided diet. The nutrition and nutrient-related pathologies, it's something so diverse, something that not only relates to genetic profile, but also to other factors. So I think actually private genetic laboratories could easily mislead the patients to believe that simply by doing a genetic test could be sufficient to individualize the diet. So that raises a very serious ethical concern that should definitely be addressed. Next slide, please. You see, for example, that the majority of the genetic laboratories in Greece, they mostly provide predisposition genetic tests, whatever on earth this means. They also touch upon pharmacogenomic testing. And interestingly, there is no information on the website whether they are actually quality accredited, if they have an ISO certification or not, which is quite awkward, if you can see. Next slide, please. And then we perform a stakeholder analysis. So basically, we want to see how the various stakeholders think about genomic medicine, how this can be implemented in Greece. Next slide, please. This was a three-tier approach. The first was to determine the policy and the content, including the goals and the mechanisms to implement this policy. The second step was to determine the players, who are the ones that they are mostly involved in the implementation of genomic medicine. And third would be to be a SWOT analysis, would be the strengths, the weaknesses, the opportunities, and the threats, the obstacles that one could face to implement this novel discipline in a healthcare setting. Next slide, please. And then you can see here that although the majority of the stakeholders are highly supportive towards the implementation of genomics medicine, you could see the press and the National Medicines Authority to have a more or less neutral stance toward the implementation of this novel discipline. But quite interestingly, you see that the Ministry of Health and the public fund, the public payer would stand opposing to this new let's say, healthcare interventions. And the reason for that, of course, is quite easy. This study was performed in 2013, which was in the middle of the Greek financial crisis, which requires a significant investment for capacity building. That's why those healthcare stakeholders would feel a bit reluctant of using these solutions in the mainstream clinical setting. This was at least the interpretation we've given. So these are the references from the papers that have been produced so far. We really expect significantly more to come out in the next years, because, as I said, that's a long-term project. Next slide, please. We have initiated a scientific journal in Greek. That means personalized medicine in Greece. In Greek, there's a website. The content is in Greek with an English abstract. Next slide, please. We have also established a national conference series. The first one was organized last year in Athens. In next slide, you're going to see some snapshots from this conference. You can see Federico Innocenti, one of the co-chairs of the flagship project for G2MC, as well as Magnus Ingemann Sunderberg from Karolinska Institute to give a talk in, as you can see, a relatively full lecture hall in Zapion Mansion in Athens. Next slide, please. We have partnered with the Golden Helix Foundation to establish e-learning courses on various aspects of genomic medicine, such as public health genomics, genome informatics, economic and health technology assessment in genomics, ethics, and so on. Next slide, please. And that's basically an overview of the courses that are offered by the Academy. Next slide, please. And lastly, we are implementing, we're using a portable molecular biology laboratory in an effort to educate and to train students, even high school students all over Greece, that is accompanied by plastic models, comics, and online games for primary school students. And the idea would be not only to educate the students of high schools, but also the educators by using a train-the-trainers approach. And this initiative has kicked off in 2016, and at the end of 2018, we got the Kafatos Lab Box Prize for the achievements we managed to get with this approach. I think that's going to be the last slide, the next one. acknowledging the players that supported this initiative so far. I must say that all this effort runs on fumes. It runs on side funding, from other projects. We are really indebted to Complete Genomics and BGI for assisting with the sequencing of a number of individuals. We really hope that in the next year to have a partnership with another big genomic laboratory. Alan is laughing. Hopefully we can have this materialized so we can manage to sequence many more of these samples we have in mind. So thank you very much for your attention. It was a machine gun pace lecture, but I hope I managed. So there is a question, the Q&A from Dr. Ligia Pereira. The question is to me is, how do we recruit participants? How much clinical data is collected? So as I said, the project kicked off in 2010. We engaged in the beginning in multifactorial diseases because we wanted to shed light into the pathogenesis of this disease, especially for Greek population. So we have established different approvals, ethical approvals for the different studies we have engaged in. So different IRB approval for ALS, different one for celiac disease, a different one for pharmacogenomics, and so on. And as such, we are entitled to collect as much as clinical information as possible along the lines of ensuring patients' anonymity. So according to all the ethical guidelines that must follow such a clinical study. Now, the plan is for the future years to have a kind of a more universal informed consent to capture all the phenotypic information, all the clinical conditions we're aiming to study. And that's actually currently ongoing, and it's going to be an observational study, of course, and that's what we expect to have by the end of this year. And I think this is going to be the easiest way of moving forward, so that we don't have to secure an individual IRB for every particular condition we wish to study. Hope I'm answering your questions.
Thanks, George. Why don't we let you go ahead and take over as co-moderator and introduce our next speaker.
So it's a great pleasure to introduce to you Gad Rennert from the Clarion National Israeli Control Center, who's going to give us an overview of the National Personalized Medicine Program in Israel. Gad, thanks so much for joining us.
Yes. Hi, George. Thank you. Excuse me for my voice. George actually laid ground beautifully to my talk. Maybe spared me some time too. And what we're trying to do is actually put the same logic or the same path of thinking into trial by using a randomized controlled trial approach. So what we want to do, we can move to the next slide, is actually test the value of the, call it personalized medicine, precision medicine approach. The value meaning that after all the inventions and all the knowledge we already have, and everything that will develop with time, we need to get it down to the ground. We need to get it to primary care. We need to make it part of regular medical service. And of course, it's very tricky. So what we're trying to do is, by way of a randomized controlled trial that I'll explain in a second, we're trying to see if intervention clinics, and again, I'll describe the intervention, overall will perform differently, better, hopefully, than the usual care clinics. And better could be on clinical grounds, on economical grounds, on satisfaction grounds. You name it, you can take a million endpoints, but we're actually comparing clinic to clinic, not a person to a person. Next slide. So we have cluster randomized clinics. we took 40 primary care clinics, 20 will be intervention, 20 will be usual care matched to the intervention by a variety of parameters, mostly socioeconomic status of the population that the clinic serves, the ethnicity of the people, a variety of other parameters. And again, as I said, it's 20, each two clinics will be compared. They'll be like in Noah's Ark. They'll go in two at a time. Next slide. And a secondary outcome will be the actually building up the genomic atlas of the Israeli population, which normally in most studies is actually the primary aim. Everybody's interested in having a full annotation of the genomic situation of the country. So for us, it's becoming secondary. It'll, of course, be collected because we will have a large population of a large variety demographically and clinically. So we'll be able to do all the work dependent on the depth of our genetic analysis. Next slide, please. So here you can see how that's the area that we're going to be functioning in. And you see that we have Jewish settlements, we have Druze, Bedouin, Muslim, Christian villages, and cities. Jews, again, we have settlements with high socioeconomic status, those with low, and those with mid, like in the middle. And actually, every circle you see is a participating clinic. One is intervention, and the other one, randomly chosen between the two, will be the usual care. Altogether, we are going to have, if we manage, it's a quite an ambitious project, as many of you can appreciate. If we are successful, then we're going to have about 100,000 participants in each arm. So the intervention clinics have about 140,000 members registered in these primary care clinics. And we assume that about 100,000 of the 140 will agree to provide DNA, provide blood. And that's based on some field experiments that we did in Israel prior to embarking into this study. So we're going to have these 100,000 whatever elements of genome, and again, like the people who presented the data before me today, and you too, George, we're all struggling for funding. It's the major barrier today is to get fundings. I gave up on formal organizations, and really one of your solutions might be mine too. And I'm looking for donors too. We're all around the, we're all after the same effort. Please note that Israel, in this sense, is very unique in the demography. We have people coming from 70 different countries to Israel into a tiny place of less than 10 million people. Really a very diverse population. We were a part of some of our populations are also known for being strong founder mutations, and quite a lot of the knowledge today in human genetics came from a lot of Jewish populations, mostly Ashkenazi Jews, although we see a lot in Sephardi Jews too in recent years. Next slide, please. So we look at the population level. That's a mandatory thing. You can call them stakeholders. You can have, that's a population. If they do not agree to play along, we have no chance. And therefore, we had workshops with people at the area of the chosen clinics. People came in, were asked about their attitudes, about the readiness to donate blood for a DNA analysis. We were a bit worried. To our surprise, practically everybody who participated in these groups until now agreed to donate blood. There could be a bias of those. We randomly invited people to participate. It was not open to the public. We chose the people we want to see, of course, randomly, without knowing who they are. And about half showed up, and all these half agreed to participate. Now, the second thing was, what do we do with the primary care clinics, with the teams, nurses, doctors, pharmacists? And of course, when our plan is to train them with measuring their attitudes before and after training, and their training will not be Genomics 101. Their training will be scenario practices. I don't want them to be scientists of genetics. I want them to know what to do with a SNP, with a mutation, with a VUS, with all these target findings. Next. Then of course, we have to undergo genetic testing. Everybody's pushing to whole genome, but let's see anybody of you do whole genome on 100,000 people. It's a challenge, and we are there. Even bigger is a challenge bioinformatics with excruciating pain to actually get the true data out of there. But we have experience in it and hope to be able to do good work on that. And then we end with the clinical action, where we need to deliver the actionable results. And again, they'll take one of two phases. One will be the identifying high-risk alleles, and the second will be the huge field of pharmacogenomics or pharmacogenetics. And we are expecting the primary care clinic team to be able to deliver it to an extent. We'll teach them the common, the usual, and of course they'll know when to triage further. Next, please. Okay, and at the very end, because it is a study, it is a randomized study, we need an outcome analysis. Okay, next slide. So that's actually the last slide. Again, status, it took us a lot of time to go through IRB, a lot of time, probably about 2 years. We already met at the population phase with 6 focus groups. At the clinic phase, we also introduced a plan to 3 clinics and started working in one where we already have 1,500 people who gave us samples. By the way, they're also giving feces samples. So about 80% of them also gave feces samples that went on to a microbiome unit that is helping us. And now we're stuck with Corona, and we're really stuck. We're trying to restart in a small clinic now, but it really stopped at this point. We, of course, are planning to go ahead once the everything comes back to partially normal situation. Thank you very much. I think the last slide should be a thank you slide. And that's our story.
Okay, thank you so much, Gadi. It's a very nice presentation. Of course, you know, you face the same obstacles as everyone does in this particular arena. And Corona just made things even worse, you know. So stick around for the panel discussion. Next talk is going to be delivered by Caterina Ferrecchio from the School of Medicine from the Catholic University of Chile. Who's going to give you an overview of the gene-environment interaction in chronic diseases. Caterina, thanks for joining us.
Oh, thank you. Thank you for the invitation to this great conference. The first slides, please. So I will present our Maule cohort. that is a population-based cohort in southern Chile. It's 200 kilometers south of Santiago. And then I will present an advancement of a gene-environment interaction that we did in this population. So here you can see this is the field work, composed by medical technologists in charge of the biobank and the ultrasound examination, and the technicians and the data managers. And you can see the red spot is where this cohort is located. Next, please. So the objectives of this population cohort is to a broad assessment of physical and mental health, and also to explore a wide range of exposures of health and healthy aging to create a biobank. It will be the first Chilean large biobank, and to follow up changes in exposure and health outcomes. And we also aim to evaluate preventive interventions of chronic diseases in this population. Next, please. As I mentioned, this is in a small town. It's a county called Molina, that is 50,000 inhabitants. And the participants are all population aged 38 to 74 years are invited. We don't do any selection, but we do have some exclusion criteria. And the target is to incorporate 10,000 participants. That is mainly the whole population that age. We began the enrollment in 2015. And we follow up then with a full examination every 2 years. And we also collect samples every 2 years. And surveillance of health events are since enrollment. And this cohort is very unique in the sense that we have a very large search for exposures, environmental exposure. This is being an agricultural county. We are very interested in pesticides and agrochemicals, but also in air pollution, soil pollution, and food contamination with mycotoxins. And when we visited them, we selected participants in their households, and we do their first assessment of the household conditions, if they have animals, domestic animals, or other type, and also the condition of the household. And then when they come to our center, that is an academic center that is located in the same area where the Molina Hospital, public hospital, is located. There we do more in-depth neurocognitive surveys, and we do a very complete physical condition assessment like speed, coordination, equilibrium, and a lot of anthropometric measurements, including bioimpedanceometry and abdominal ultrasound. We count the teeth, and we do a peak flow test. And then we collect blood, urine, and saliva samples for biobanking. And in the subsample, we also collect stools. Next, please. Well, this is just a short image of how the biosampling proceeds. From the health center, we take the first samples, and they are kept at 4 degrees centigrade until moved to the center of the hospital, where we have our transient biobank. And there the technician proceed to create the aliquots for each participant, and storage at -80 degrees centigrade. And every 3 months that I move to the final biobanking in Catholic University in Santiago. All the procedure follows the biorepository identification system of NIH, so we follow the same. Next. So in this population, we already have almost 10,000 people enrolled. but with the first 4,000, we share the white blood cells with the Dr. Ricardo Verdugo from the University of Chile, who extracted DNA and did some polygenic studies. So on here I will present the result of one of our areas of research, that is gallbladder cancer and gallbladder diseases, because Chile, and in particular Chilean women, have had for many, many years, for decades, the highest worldwide rate of this cancer, and also one of the highest rates of gallbladder diseases and gallstones. So one of the things that the government have a very active and aggressive program of gallbladder extraction, cholecystectomy, and there are some evidence that cholecystectomy may cause very, very deep metabolic problems, including non-alcoholic fatty liver disease and hepatic cancer and other digestive cancers. So what we explore here is the role of a polygenic risk score for non-alcoholic fatty liver disease and interaction with the cholecystectomy. We also did the role of obesity, but I will not present because we don't have time. Next slide, please. So this is just to show you that the first reports of this association was in 1984, and since then have been a lot of reports of this association of cholecystectomy and non-alcoholic fatty liver disease. And the last is ours, that we just submitted recently to the American Journal of Gastroenterology, in which we found that this association was stronger and significant in men more than in women. Next. So here is the summary of what the methods used by Ricardo Verdugo's group. So he did two things for us: the evaluation of admixture using 150 SNPs for this categorization, and also did the non-alcoholic fatty liver disease with 9 variants for this disease. The latter is based on the European and North American scores, risk scores. Next, please. So what he found is that, so in the top panel, we can see the distribution of our participants, the ethnicity. So the most important are in green are the European, and in orange the Mapuche Amerindian. We have in the northern Chile is the Calypso, the light blue, that is the Aymara that we share with Peru and Bolivia. And the orange, the Mapuche, which is southern Chile, and we share with Argentina. And what is interesting, in the lower panel, it shows the non-alcoholic fatty liver disease cases in red and the controls in green. And the only panel that shows higher diversity, increasing the red column as the polygenic risk score increases, is in Europeans. As the Europeans go down, their association gets poorer. Next, please. So when we saw the role of cholecystectomy and polygenic risk score, this is summarized in this table, and the denominators is 17 and 1600 in each cases and controls. So if you look at the first row, it shows the 4 quartiles of the polygenic risk score from lowest to highest. And you can see when you have no cholecystectomy, that is the first 5, practically no effect, at least no significant effect of the polygenic risk score. But when you have cholecystectomy, you see increase in risk, very significant since the second quartile of risk. And interaction synergy index is positive, showing an interaction of the polygenic risk score with the occurrence of cholecystectomy. So having these high risk SNPs puts you at higher risk of developing the non-alcoholic fatty liver disease once you have a cholecystectomy. And this is very new. We are just analyzing this data. We are very excited of this result. Next slide, please. So just summary, the discussion is that so what we found, we confirmed the risk of non-alcoholic fatty liver disease associated with cholecystectomy increases with the increasing polygenic risk score. Since polygenic risk score was better predictor of non-alcoholic fatty liver disease in people with higher European ancestry, we hope that if we had this SNPs discovered in our population, probably the prediction will be better. So this is something that we think we will be able to do soon, thanks to the collaborator of one of the nice USA-based companies that will collaborate with us in do a full exome mapping in the whole cohort. That will be the huge assessment for my country. My last words is I want to mention that since COVID epidemic arrived to my country, and we have this cohort, we decided that we should go into MAUCO COVID project. MAUCO as a sentinella cohort for this pandemic is ideal. So we began with a series of sequential rounds of surveys of the experience of our participants with the COVID disease. And we already had this will be 1,000 participants every month to get like the temperature of what's going on in the population. We hope to be able to incorporate very soon biological sampling to these surveys for serology, molecular testing, and the biggest aim is to do a full genomic analysis of the host and the virus. And last, please. So this is just to introduce you to our research center. It's the Advanced Center for Chronic Diseases. It's a state-funded research center in which the two largest research university in Chile, University of Chile, that is the state university, and Catholic University of Chile, joint efforts to do this. And we go from epidemiology and public health, that is my field, to basic sciences, that is more in the University of Chile part. And thank you very much, and I'm sorry for the problems with my microphone.
Thank you so much, Katarina. It's a great pleasure being with us today. Thank you so much for joining, Aymar, and look forward to your talk about polygenic risk score for common diseases.
Great. So it's a great pleasure to be here. Thank you to the organizers for inviting me, and also this is it's been a pleasure to listen to some of the talks. This is a very exciting group of speakers and really focused on very important problems. So the topic of my talk today is really following on from the previous talk, is also focused on polygenic risk scores and the challenges and opportunities for calibrating polygenic risk scores and improving their performance in diverse populations. Next slide, please. So just before I start, here are my disclosures. Next slide, please. So it's a very exciting time to be in the field of human genomics. We have a technology that's one of the most powerful technologies that we've ever built. The cost of genomes have plummeted, and there are hundreds petabytes of genomic information now in 2019, 2020 being produced yearly. So it is not an exaggeration to say that we are an era where we have information on hundreds of millions of genomes in humans. Next slide, please. And this has meant that our knowledge of the genetic architecture of disease has really exploded. And we have interrogated genomes for, at this point, thousands of diseases and biomarkers and other human traits. This explosion has particularly happened in the last couple of years, where we've really seen the numbers and breadth of disease studies explode. Next slide, please. And a lot of this knowledge is now being used beyond research and beyond understanding the biological underpinnings of disease, also being brought into clinical practice in ways that are now well established for diagnostics for genetics disease or for family planning, and in ways that are really emerging using genomics for screening for preventive health and for pharmacogenomics. And also there are market forces at play, and direct-to-consumer offerings are a very big industry and growing. Next slide, please. Thank you. But we as a field are going to need to face some challenges in the coming decade. This one is one that is the subject of my research, and I think is something that is of interest to many participants in this meeting, which is that genomics is failing on diversity. By that I mean that the vast genomic databases that we have built are depleted for information for the vast majority of human genetic diversity, and indeed for some areas of the world really absent for human genetic diversity. And we are relying our interpretations on databases that are enriched for European ancestry individuals. So it will be important to understand and evaluate what the limitations of those databases are, as we also pursue strategies to rebalance those databases with representation. But it will be important to do both in the next 10 years. Next slide, please. So this is a study that we published in my group a couple of years ago in the American Journal for Human Genetics, where we looked at the impact of taking polygenic risk scores that have been predominantly constructed using data from one population, in this case using data from European population. And if you just take that risk score and naively try to use it in other populations, we were able to show that the genetic prediction accuracy decays with increasing genetic divergence between the discovery and the target population. We went on to do extensive simulations of demographic data, and we were able to explain, at least theoretically, why that might be the case, that the biases were actually linked to the majority of variants in those models really be under forces of genetic drift. Sorry, that's my alarm. And that neutral human evolution was sufficient to explain the difference. So this offered us a theory, at least, about why we would see differences just transferring polygenic risk scores across populations. Next slide, please. So in 2020, if we, as I mentioned before, if we want to grapple with some of these challenges, we really have to identify what are the key questions that we need to face when we think about integrating genomic risk information, not only into biological research, but as I discussed, how we're using it now to improve health and wellness. And some of the key questions, in my opinion, are how we transform genetic research into medical knowledge, and on acknowledging and measuring what are the biases in our current knowledge bases. And then once we have identified genes that are clinically relevant, how do we then systematically identify individuals and communities and populations who harbor those risk or sometimes protective factors, and what other factors are at play over a patient's life course that determine health outcomes. And then a question that I'm also very interested in, it's not the topic of my talk today, but it's certainly a big challenge to face, is how do we integrate this information more broadly in health systems and arenas of medicine where people are not necessarily genetic experts to improve healthcare management. Next slide, please. So what I wanted to talk about today are results from a study called the Population Architecture Using Genomics and Epidemiology, or PAGE, P-A-G-E, which was really a study that followed on from the paper in the American Journal of Human Genetics, where we theorized why polygenic risk scores might not translate across populations. The nice thing about the way that the PAGE study was designed is that we could really test that theory. So before I talk about that, I really want to acknowledge that a very, very talented postdoc who's now an assistant professor at Johns Hopkins, Jen Wojcik, led this work. This work was published in the journal Nature last summer, but it really comprises work from very many investigators, some of whom are also on this slide, and many sites across the US, including Mount Sinai, including Fred Hutchinson, the University of Southern California, and University of UNC Chapel Hill. Next slide, please. So the design of PAGE was really to investigate ancestral diverse populations to understand, improve our understanding about how genetic factors influence susceptibility to disease. Our focus, because this is a US-based study, was on minority populations in the US. We started this study at this point nearly 7 years ago. And at the time we started the study, this is one of the largest uniform cohorts of non-European populations. So it comprises about 17,000 African Americans, about 4,000 to 5,000 Asian participants, about 22,000 Hispanic Latino, and about 4,000 Native Hawaiian Islander populations, a couple of hundred Native American populations, and some folks who don't necessarily fit into one of or those particular categories. Still today, this still represents even 7 years later, a very large data set collected and genotyped on a standard platform. Next slide, please. So I want to walk through some of the challenges that you encounter when you sample for enriched diversity in a cohort, and some of the strategies that we developed to that helped us empower our analysis. So one of the first challenges when you sample specifically for diversity is that you start to lose discreteness in terms of defining population groups. And by that, I mean genetic ancestry. So although people may self-report, if you ask the question about race, ethnicity in the US, particular ways, if you actually look in genetic space, what you see is a continuum of genetic ancestry representing ancestry from the continents of Africa and Asia and the Americas and Europe, which reflects the history of population groups in the US. And you see this not only at a continental level. If you were, for example, to also take self-reported Hispanic Latino populations from across the US and look at subcontinental ancestry, you will also see a continuum of ancestry representing the continental Americas versus, for example, the Caribbean. Next slide, please. And it was really that early observation that encouraged us that to not think about separating population groups, at least for the PAGE consortium, rather develop strategies which included developing a microarray that works across all populations equitably. It includes developing statistical tools that would enable for joint analyses across populations, and allowed us to do a GWAS of jointly across populations in a parsimonious way that accounted for allelic heterogeneity across populations and even differences in variance at alleles across populations in the statistical model. And so in the PAGE study, we have collected hundreds of phenotypes. We focused on 26 for this first paper that we published. And not surprisingly, we made very many discoveries. Most of the traits that we focused on, which are largely cardiometabolic, although there is some lifestyle traits in there and some kidney disease traits, most of these already had very, very large, predominantly European studies available. So it won't be surprising that many of the things that we discovered in our study were already discovered in the GWAS literature. So we were now replicating many of the associations that had previously been discovered. Nevertheless, we were able to make some novel discoveries. So we had 27 novel associations across 26 traits and 38 secondary associations. So when you condition on a known variant, you still see a significant association at the locus. It won't be surprising to many of you to know that this is because those variants are rare or absent in European populations. So we're really in this design enriching for variants that you see specifically in non-European populations. Next slide, please. But the other thing that we could do, which gets back to this question of the transferability of polygenic risk scores, is we could take that set of variants that does replicate across European populations and other populations, and there were very many. There was over 1,400 of those loci, and then we could ask at those loci that transfer across population, what's going on with polygenic risk scores? Next slide, please. And so this is what we discovered. So when we took variants that we replicated from European studies and looked at the effect size of those variants in African American and in Hispanic Latino populations, we saw that the effect sizes were weaker in the other populations compared to the target population. And what we deduced from this is not that the biology or causal variants are different across populations, but the way that those variants are tagged by the variants we're testing might be different. And that is what is impacting our ability to use those variants for things like risk prediction. Next slide, please. Another way that we could look at this is we could look at the impact of diversity in genetic GWAS, genome-wide associations, to improve our understanding of the potential causal variants. So in this exercise, what we did is we took a very large European data set and we meta-analyzed it with the PAGE data set of diverse populations and compared it to a similar meta-analysis of a same-size database, but this time taken from the UK Biobank, downsampled to 50,000 British-born white participants. And we could see that our top posterior probability at hundreds of loci for height in this example was improved when we meta-analyzed the large GWAS using a diverse data set compared to a homogeneous single population data set. Next slide, please. And we can dig down into the reasons for that. So if we go and take an example locus from that association and we do the side-by-side comparison, so on the left-hand side, we're looking at this locus in the UK Biobank meta-analysis, and on the the right-hand side, and on the left-hand side, we're looking at this analysis with the page meta-analysis. You see the locus zoom for the association on the top level, and you can see the magnitude of association is very similar, slightly higher actually, with a lower p-value when you meta-analyze with the UK Biobank, but the locus is still significant in both analyses. However, when you go to the middle layer, you see that your resolution of the putative causal variant at the mapping interval is stymied and low-powered in the UK Biobank meta-analysis. In the page meta-analysis, we are actually able to resolve a single credible SNP at the interval that we believe is underlying the signal. And the reason for that is explained by the last layer of this plot, which shows you that in European populations, those SNPs at the mapping interval are all in tight equilibrium, so that we have no power to distinguish the credible leading SNP, whereas in African American and Hispanic Latino populations, the LD, the linkage disequilibrium architecture is different, so now we have power to distinguish which is the causal variant. Next slide, please. And you know, I think just to finish up, that we also need to think not only about the impact of LD architecture across populations and how we're using genomics to predict risk scores, but also all the other determinants of health, including environmental and behavioral determinants and social determinants of health, and also sometimes evolutionary forces that are acting on the genome in different places in different parts of the world. So I will leave you with this example, which is close to my heart because it was a project I worked on when I was a postdoc at Stanford University, where we mapped the underpinnings of blonde hair in the Solomon Islands in Melanesia, which was really caused by a single missense amino acid change in the gene TYRP1 that had not been discovered in European populations, even though there were very large studies of hair color at that point, even 10 years ago. And because it was quite rare in European populations, but quite frequent in Melanesian populations and really driving hair color in certain Melanesian populations, it was quite pleasing to me back last summer when the first tranche of exomes were released for the UK Biobank, that they posted on their blog that they finally, using a tranche of data that was 50,000 persons deep, started to see the same variant in the UK Biobank, and that it and many other very rare variants in that gene were linked to blondism in the UK population. So that was very pleasing and makes a lot of sense biologically, but I will point out that that study design required exome sequencing in a very large number of people because variants are rare in that part of the world, whereas in the Melanesian study, it was really less than 100 individuals in our study design because that variant was quite common, and the architecture of hair color for blondism in Melanesia was much more simple to understand. Next slide, please. So I'll just finish up with some notes of optimism. While I think I've pointed out some of the challenges that we still face incorporating population diversity into research, I think I'm also pointing out, and I think many people here would agree, that it is imperative we do so. And there are many opportunities and benefits of doing so. And these opportunities really have the potential to transform medicine and even society as we know it today. Genomics does tell us about our susceptibility or protection for disorders and diseases, but it also tells the broader story of evolution and our ancestry and our history, which in turn can be correlated with social environmental determinants of human health. And we need to factor those things into the picture and the genomic engines that we're using. I think this is both a challenge and opportunity, but it is very important to embrace to get a fuller clinical and social picture of health and wellness. Next slide, please. With that, I'm just going to thank my colleagues and collaborators and point out particularly Joan Wojcik, who led the analysis for the PAGE study with Christian Yu and Gillian Belbin, and Alicia Martin, who led the analysis in the American Journal of Human Genetics paper I described, and all the collaborators in the PAGE study. Thank you.
Terrific.
Okay,
Thank you so much, Emer. Really terrific summary of important information that touched upon actually all of our prior talks. So we're a bit over time, but let's move into the panel discussion, and certainly we welcome any questions. So I have a question to Gad, and I continue to be really fascinated with your idea of essentially a randomized clinical trial of genomic medicine. Essentially, you're not making the supposition that implementation of genomic medicine will be good, and you want to prove whether it will be good or not. Most clinical trials, traditional ones at least, have a data safety and monitoring board that monitor progress and ultimately and see preliminary data and can actually make judgments about whether to stop the trial early because of efficacy that's proven in the intervention group, that the control group from a ethical point of view shouldn't be denied. How are you going to handle those kinds of situations in this modern genomic medicine randomized clinical trial?
Okay, a very good question, Alan. The truth of the matter is that because our, first, of course, we'll have the algorithm committee. The reason we do not have it in place yet, and we're missing some more, let's say, formal bodies that a study of that magnitude deserves, is that I'm still, I wouldn't say hesitating, but I'm still struggling with actually seeing how feasible the whole study will be. Because you understand that the cost of the study, even beyond the genetic testing, is immense. And we're constantly approaching more and more bodies to actually get money to establish it to the extent that that it's a fact. Although I probably am going by the end of the month to have 2,000 participants already signed on IRB-Fund. The issue with ethics is that my control clinics are usual care. No data is withheld from them. Every genetic finding, if we test BRCA, and we made BRCA a national screening now in Israel for breast cancer, the usual care will do the same. So we're not withholding any service, i.e., we're only trying to see whether knowing more genetics and putting it into action actually helps more. So, because we're kind of sold to the subject, we all think that this is the case. We should do it. And of course, we all have these specific single examples that no doubt show the benefit. My issue is to look at it as a whole, because if it does work as a whole, then the health systems have to start incorporating it into their daily activity. That's not the case today.
We do have a hand raised from Dr. Rupesh from Nepal, and I will go ahead and allow you to talk, Dr. Rupesh, and you can unmute yourself. Go ahead.
My question is to Dr. Zoz. Sir, as you have shown some of the conditions, like genetic conditions, and you have gone for whole genome sequencing, rather than going for whole exome sequencing, I would like to know what were the mutations, and they were in the exome region. If they are not in the intron or in the third prime or regions, then why don't you do the whole exome sequencing rather than going for the whole genome sequencing?
Mm-hmm. Well, as you know, whole exome sequence just captures 3% of the genome. And in the case that we want to address multifactorial diseases and we go for association studies, then in this case, it's absolutely necessary to try to have a complete picture of individual genomes. So that's why, and we opted to look at whole genome sequence to capture all the information we needed. Of course, when we aim to explore the pharmacogenomic variants, we only restricted ourselves to a proprietary panel of genes that they are of pharmacogenomic interest. Of course, it's quite appealing to us to try to get the genetic composition of variants that are not related to the genes that are mostly mutated. For example, if we want to look for beta thalassemia mutations in genes in Greece, then you don't only look at the globin genes, it's also quite interesting to look at other genes. And in this case, again, you should look by whole genome sequencing. And at the end of the day, you know, if the cost of whole genome sequencing cost, it's actually threefold higher compared to the whole exome, then it's really worth looking, you know, investing a bit more and looking into the whole genome aspect rather than whole exome.
Okay, thanks for your answer. Thanks.
I can ask a question of George. George, I really enjoyed your comments about personalized nutrition and the possibility that genetics could determine some aspects of that. And I think all of us were thinking the fact that in Greece, we all hope or wish that we had as healthy a diet often than the Greeks practice in terms of the so-called Mediterranean diet. If you did come across genetic markers that suggested something other than a Mediterranean diet was healthy for a given individual, how adherent do you think the population or those individuals would be?
Well, we have been quite adherent to the COVID-19 measures, so we did pretty well. Well, actually, seriously, I really find myself sometimes in a very awkward situation because I'm really a very strong opponent of this particular discipline, and I really feel sometimes myself I will find myself in a river with cement shoes. We have a lot of laboratories that are selling this type of test to the patients, and many people are buying for it, and they really feel that's for lifestyle. Some others feel it's really beneficial. Actually, strictly speaking, SNP-based genotyping to determine one's diet, it's simply not enough. It's not enough. You need to take into consideration other parameters. I'm not talking about environment. One thing you should take into consideration is the gut microbiome, which is quite important. The other one is transcriptomics. So simply by performing association studies is not going to work. It will lead, you know, the patients and the dietitians nowhere. It's very sexy. It's very trendy. I understand this. It has been in the laboratory, you know, portfolio for many years. There has been a mass opposition in our paper that came out 5 years ago that all this by doing a meta-analysis of over 2.5 million patients and control individuals that such thing as DNA variant-based prediction of nutrient-related pathology, it's simply it's not feasible, right? And we had some very strong opposition by private laboratories in the US, having banned one of my interviews in you know, New York Times, just because I just said so, right? Or applying to the journal to retract the manuscript. So it is a big market. Yes, I have all this on paper. So it is a big market. There's a big financial interest. But we are obliged to protect the public and the patients. That's how I stand.
Katerina, if you're still there and can take yourself off of mute, question about how representative the Marco population is to the greater Chilean population, and secondly, a question about interactions between pesticides and potentially neurodegenerative diseases like Parkinson's.
Yes, well, regarding the comparability with the Chilean population, this is a population, well, genetically is very similar to the average of the distribution of ancestry in the country, very similar to the Santiago ancestry distribution. But this is a lower socioeconomic level and have more probably environmental exposures or different than those like say in Santiago. But it's similar to the vast majority of agricultural towns in Latin America and Chile. And regarding the interaction with pesticide exposure, yes, this is an area of research. We have a colleague working on this for some years, raising the information about the exposures and also collaborating with some European colleagues to do the measurements of not only pesticides, but diverse agrochemicals that are used during the year. So we are doing like a surveillance of environmental exposures in the fourth season of the year, and also related with the, so we, among the population, is with like 40% has worked in agricultural or is working. So we split the population, the participants in direct exposed or indirect exposed, environmental exposed, or work-related exposure. So we think we will have a very good exposure assessment for pesticides, but still we are in the collecting phase of the studies.
Great, thank you. And maybe one last question that actually could be a symposium in and of itself, but maybe some very short answers from the panelists. Maybe starting with Emer. So what's the bottom line? What do you think about use of the PRS in clinical practice?
Yeah, so I think there's enormous enthusiasm for using polygenic risk scores in clinical practice. And really, I think in the last few years, we've started to see the ways in which polygenic risk scores can be used. So for example, one application that I've seen quite useful is if you take people who have monogenic risk for disorders that we include in population screening programs or incidental findings from diagnostic testing, for example, for certain cancer conditions or cardiology conditions. If you have cohorts of people who have existing monogenic risk, and then you also combine polygenic risk, then you start to see, you start to get a little bit more predictive about people's monogenic risk in the context of their polygenic risk. So that's one application certainly that many people have been interested in developing, but even polygenic risk itself, using these type of algorithms in healthcare, the way that we think about using monogenic risk for preventive health. There are companies now with offerings. There are places in the world that are starting up clinical centers that would offer these types of testing. People often think of me as being a bit of a downer on it because I tend to point out problems with polygenic risk. I am very confident about our ability to resolve these problems and improve these tools that we're using. And I think there's an enormous amount that we need to learn on the clinical side. In addition, and the fuller context of how these tests may be useful, and in some cases maybe they won't be all that useful, but undoubtedly I think that they will be. Actually, I just came in at the end of Dr. Furicchio's talk, and I think NAFLD is definitely an area where there would be a lot of utility if we could get good polygenic risk scores.
Terrific. Well, I'm going to have to cut it off here. I'll ask George if he has any parting remarks before we turn it back to Gabriella and Bruce.
I think we're well over time, so for the sake of time, I think we better turn it off to Bruce and Gabriella for ending the first day of what was actually a quite successful virtual conference.
Gabriella, do you want to start?
So yes, first, can you hear me?
Yep.
Yes.
Yeah, great. So first of all, I want to really thank our speakers for a great world tour of a lot of things that are going on the genomic medicine landscape. We heard about great national initiatives, other initiatives on specific populations, specific diseases and tools such as pharmacogenetics and cancer. And I think that what we learned about the relevance of patient involvement was also really crucial. And this concept of co-production that Cecilia explained to us today, that was, I think this is really a very relevant consideration in all our implementation work. And I'm also happy to see that a lot of emails have been exchanged through the chat, which I hope will really become an opportunity for broader international collaboration. Turn it back over to Bruce.
I'll add my thanks. I think, we still have 150 odd participants, which is remarkable. And just want to thank the speakers, moderators, and the participants for their involvement today. I guess the one thing that impressed me, looking across the kind of spectrum here, is there's a lot going on. A lot, honestly, for me at least, in places I had no idea what was happening, and I think it was eye-opening to hear some of the initiatives. And I guess the one thing that will be interesting to track, and I think it gets to the point made by Emer just now and many others across the day, is that we really need to look at what the reach of these studies will turn out to be. That it may be the case that lots of countries have initiatives, but then we have to look internally as to how diverse the populations are that are actually participating and serving. And I think this will be a really important area going forward if we're going to really see genomic medicine reach the potential that it has for large populations. So I hope that G2MC maybe can help to track some of this progress as we go along. That is really all I had planned, and I know it's crazy hours of the day for some people. Any final comments from Hannah or any of the organizers?
Yes, so just to reiterate, every question that was put in the Q&A that may not have been answered will be delivered to the correct speaker. And then every chat is also being captured, so we do have all that information. And then just anything else, Gabriella and Bruce?
I wanted to thank you and Teddy and Daniela for great work, and everybody else that's behind the scenes. We couldn't have made it without you, so thank you.
I would
Thank
like
you.
to say thank you to Hannah and Daniela for their hard work in organizing this, and thank all of the panelists and our attendees for joining us. We actually at one point had close to 200 attendees, all totaled. So hopefully we can have that kind of interest tomorrow too. So thank you very much.
Thank you. Bye-bye.
See you all tomorrow.
Bye-bye, see you tomorrow.
Bye-bye.
See you tomorrow.